Hey there, my name is Jeremy Martin, and today we'll talk a little bit about glycolysis, gluconeogenesis, and aldolase in its function and expression. So, in that, we'll be discussing the process of glycolysis, which is a sequence of reactions which produce ATP, NADH, and pyruvate from glucose. We'll get into gluconeolysis. Uh, let me rephrase that. Glycogenolysis, which uses the stored reserves of glycogen polymers to restore glucose concentration. We will also get into gluconeogenesis, which also creates glucose, but more so in the reverse process of glycolysis with a few different steps. Gluconeogenesis will use pyruvate or oxaloacetate derived from amino acids. Then we'll also learn about fructose bisphosphate aldolase, more commonly known as just aldolase, and its responsibility for fructose metabolism, as well as the regulation of specifically aldolase B expression. In conclusion, we'll also discuss the result of genetic defects to the gene for aldolase B, commonly called hereditary fructose intolerance. Starting off with glycolysis, this takes place in the cytosol of cells and does not require oxygen. It can be literally broken down into glucose splitting, glyco being glucose, lysis being splitting, as it occurs after someone eats and is in their fed state with plenty of glucose newly added to their digestive system. Glycolysis has an ATP investment stage and an energy payout stage, which occurs the payout occurs once for each compound produced by the investment stage. Beginning here with that investment, hexokinase, which I will later call glucokinase, uses one ATP to phosphorylate glucose into glucose 6-phosphate, which cannot leave the cell as it becomes phosphorylated. The isomerase modifies glucose 6-phosphate into the similar fructose 6-phosphate molecule. Phosphofructokinase then uses that second ATP investment to phosphorylate the fructose 6-phosphate into fructose 1,6-biphosphate. One good way to remember where the ATP is used is anytime a kinase is involved, it's using an ATP. That fructose 1,6-biphosphate can then be broken into dihydroxyacetone phosphate, which I'll now call DHAP, and glyceraldehyde 3-phosphate, or G3P. This happens because of that aldolase enzyme. DHAP can be modified into G3P by the triose phosphate isomerase and vice versa as needed. In the second part of glycolysis, G3P proceeds to the energy payoff phase. So DHAP is constantly converted to that G3P to maintain equal concentration between the two compounds. In other words, one glucose molecule will produce two G3P that each continues into the following redox reactions to produce a net gain of 4 ATP, 2 pyruvate, and 2 NADH between those two G3P molecules. So in this process, number 6 here tells us that G3P is oxidized and NADH plus is reduced to NADH and a hydrogen ion. Using this energy from the process to phosphorylate G3P to 1,3-bisphosphoglycerate. The 1,3-bisphosphoglycerate then donates one of its phosphate groups to ADP to create ATP, turning it into 3-phosphoglycerate in that process. An isomerase in number 8 here, then is going to convert the 3-phosphoglycerate into its isomer 2-phosphoglycerate. Number 9 then tells us that this molecule loses a molecule of water to become phosphophenylpyruvate, which I'll now call PEP. To finish glycolysis here, that PEP molecule is very unstable, so it readily donates its phosphate group to another ADP, which turns PEP into that outcome pyruvate. Remember, those steps happen to each of the G3P created during the investment stage. So this final process actually creates 2 ATP, 2 pyruvate. With the NADH and ATP created earlier in the payout stage, the net gain is, again, 4 ATP, 2 NADH, and 2 pyruvate. Pyruvate goes on to be used in the citric acid cycle to produce more NADH and ATP, or onto gluconeogenesis, where it is converted into oxoalaacetate for its journey back toward the beginning of glycolysis, glucose. When someone eats a meal, some of the glucose is processed into glycogen. This occurs when a large source of glucose is available. To begin the process, that hexokinase or glucokinase involved in glycolysis is also responsible for creating G6P with a cost to ATP. G6P is altered to G1P, which in short is linked together by branching enzyme and glycogen synthase to produce that glycogen polymer. Glycogenolysis then occurs after glucose concentration is exhausted, usually after about 8 hours of fasting. The breakdown of glycogen to glucose might end at G6P, which can be used in glycolysis and removes that initial investment step in phosphorylating the glucose. The 2 enzymes unique to degrading glycogen are those which help create the G1P, called glycogen phosphorylase, which regulates the rate of glucose released from glycogen, and debranching enzyme, which is responsible for removing those segments of the G1P, which can be altered then again into the G6P. Glycogenolysis occurs beyond that rough 8 hours of fasting when liver glycogen stores start to deplete and an alternative source of glucose is required. It occurs mainly in the liver and the kidney cortex. It is a metabolic pathway that results in the generation of glucose from non-carbohydrate carbon-containing compounds. There are 3 main precursors. Could be found in lactate from the anaerobic glycolysis in working muscle and red blood cells. Could come from glycerol, which is released from adipose tissue breakdown of those triglycerides, and pyruvate and amino acid, which are both used to form that oxoalloacetate. Lactate and glycerol can be processed to form pyruvate as well. Glucagon hormone presence results in the inhibition of phosphofructokinase 1 involved in phosphorylation of fructose 6-phosphate. It also activates fructose 1,6 biphosphatase, which does the opposite then. Simultaneously as that, pyruvate kinase is inactivated, preventing the dephosphorylation of PEP, while pyruvate carboxylase becomes activated to then start forming oxoalloacetate from the pyruvate. Gluconeogenesis has a close relationship to glycolysis. While glycolysis is the breaking of glucose, gluconeogenesis is the creation of glucose, literally meaning glucose new creation. However, gluconeogenesis is not as simple as simply reversing glycolysis, as there are some irreversible steps in glycolysis. The enzymes unique to gluconeogenesis include pyruvate carboxylase and PEP carboxyl kinase, which convert pyruvate to oxoalloacetate. And then PEP. Fructose 1,6-biphosphatase is also important in converting fructose 1,6-biphosphate to fructose 6-phosphate. Finally, glucose 6-phosphatase turns glucose 6-phosphate on up to that final resulting glucose. Again, the first step in converting those two pyruvate molecules into two molecules of oxaloacetate requires the hydrolysis of 2-ATP by the pyruvate carboxylase enzyme. So we're using some ATP, unfortunately. Then PEP carboxykinase enzyme converts two molecules of the oxoalloacetate into two molecules of PEP with hydrolysis of two GTP molecules, which again is just the guanine version of ATP. Later in the cycle, that F16BP must be dephosphorylated to F6P. And after a similar isomer change, the final reaction is also unique, as that G6P phosphate is also dephosphorylated. Again, that final glucose. Also known simply as aldolase, fructose bisphosphate aldolase is very important in the metabolism of fructose sugar. Particularly, it is involved in both glycolysis and gluconeogenesis as the catalyst to convert F16BP to DHAP and G3P, or vice versa. Remembering that DHAP is either used to form triglycerides for storage or is converted to G3P as needed, which is then required to proceed into that payout stage of glycolysis. In short, this enzyme is vital for effective energy storage and production, no doubt. There are three isozymes that occur in mammals. Aldolase A is found in muscle and blood cells, active in glycolysis in these tissues. Aldolase C is found in brain tissue, also really just used for glycolysis in this tissue. Aldolase B, however, is a special enzyme variation found in the liver, kidney cortex, and a little bit in the small intestine tissue. This enzyme is unique because it's active in glycolysis as well as gluconeogenesis. The aldolase B protein is coded for by the ALDO-B gene found on chromosome 9 in humans. There are a few transcription factors that can determine whether this protein will be processed from DNA to its active form. Each factor has a slightly unique way of initiating or inhibiting transcription. Hepatocyte nuclear factor 1 and deboxing binding protein are strong transactivators of aldolase B, which means the gene expression will be increased as they bind to their designated promoter region site. That first one, shortened as HNF1, however, shares a similar binding site as HNF3. Therefore, HNF3 competitively actually inhibits HNF1 when bound. When present, although not being non-competitive to DBP, it is still prevented from binding, which means HNF3 decreases the expression of aldolase B anyway. The final regulator is C enhancer binding protein, which is also found to strongly activate that aldolase B construction in liver cells specifically. It's been found that an abnormal increase in expression of aldolase B can result from a greater intake of carbohydrates, releasing a decreased concentration of glucagon hormone. With less glucagon present, glycolysis will occur more frequently than gluconeogenesis. Hereditary fructose intolerance is an autosomal recessive disorder, typically reducing the amount of aldolase B produced. There are more than 30 mutations identified to be associated with the disorder, most of which can be inherited, hence the name. The result of altered aldolase B enzyme is a buildup of fructose 1-phosphate, which prevents the creation of G3P and DHAP. With less G3P available, these individuals lack the intermediate needed for the payout stage of glycolysis. Therefore, they are considered hypoglycemic. Furthermore, the excess of F1P holds the phosphate groups in a sort of reservoir, making them unavailable for ATP production. Similarly, in the opposite direction, phosphate groups are also not available for gluconeogenesis to undergo the needed phosphorylation reactions to result in some more glucose formation. Overall, the altered aldolase B results in less ATP production and therefore inhibition of protein synthesis in the liver and kidney. This can lead to tissue damage or even organ failure. In summary, glycolysis is the series of reactions and isomer changes that result in a net gain of 4 ATP, 2 NADH, and 2 pyruvate. Glycogenesis involves the creation of a glucose from a glycogen polymer of branching glucose 1-phosphate chains. Gluconeogenesis also results in glucose formation, but from pyruvate and amino acids generated from lactate or triglyceride conversion in liver tissue and kidney cortex specifically. Aldolase is responsible for breaking down fructose to G3P and DHAP, which can be used for glycolysis, or DHAP can go on to be stored as triglycerides in adipose tissue. Aldolase comes in the A, C, and importantly, the B form. Regulation of aldolase B expression is done by transcription factors, which must bind to a promoter site on the DNA to initiate the process. The HNF1, C/EBP, and DBP activate aldolase B transcription, while HNF3 inhibits this process. Finally, we discussed the effect of altered expression in the enzyme. In those with high carbohydrate diets, aldolase B expression increases and glucagon hormone presence is then reduced. Those with reduced aldolase B expression are often diagnosed with the genetic disorder hereditary fructose intolerance. An altered DNA sequence for the protein ultimately results in less ATP production and hypoglycemia, both of which can cause liver and kidney damage or failure if not managed properly. Here's a list of some references, some image references as well. Take care.